Hey guys, welcome back. We have finished carbohydrates and we are on to lipids. So, our next major class of organic molecules that make up living things are lipids. So, lipids do a lot in our bodies. The first is they store energy. They are a major energy reservoir and they also form the structure of cell membranes. They're weighing in at about 9 calories per gram as opposed to carbs and proteins 4 calories per gram. Lipids are pound for pound the most energy you can get out of a food is a fat. All right, so lipids are fatty, oily, or waxy organic compounds that are insoluble in water. Fats do not dissolve in water. Olive oil does not dissolve in water, that kind of stuff. So there's a lot aside from just like fat on animals and humans that are made up of lipids. Triglycerides are made of lipids. Phospholipids are also incredibly important. Waxes for plants and some animals are very important. And then actually steroids are also a type of lipid that are going to have huge effects on biological organisms. So a lot of lipids are going to incorporate a fatty acid. We've seen this guy before. He's a pretty simple organic compound. They've got carboxyl groups that are joined to a backbone of carbon atoms. Could be as few as 4, could be as many as 36, but a lot of carbon atoms. And so we've got two types of fatty acids I want you to know about. Saturated fatty acids are generally going to be found in animal fats, and these guys have only single bonds. When you only have single bonds between your carbons, you have as many hydrogens as you can possibly have. You're saturated with hydrogens. So these molecules can pack in super tight and they're solid at room temperature like an animal fat would be. There are a few fruits that have saturated fats, but they're pretty rare. Unsaturated fatty acids are vegetable oils and are plant-based stuff. These guys have one or more double bonds. Now, as you start to throw double bonds in carbon chains, you have fewer hydrogens than you could potentially have bonded. So, we call it unsaturated because it is not totally saturated with hydrogens. It puts these kinks in molecules, and they tend to not be able to as tightly fit together. So they're generally liquids at room temperature, and they're quite a bit easier to digest for us as well. So there's unsaturated and saturated fats. You'll hear that in your everyday kind of conversations about foods, and that's really all it means. So here's our saturated and unsaturated fats. Saturated fat, as many hydrogens as we could possibly put on here are on here. Here's linoleic acid, so we got a double bond here. Well, now we're missing a couple hydrogens. And a double bond here, we're missing a couple hydrogens. Now, if you're an enzyme, you got a lot of space to get in here and work now that you didn't have before. So this is an unsaturated fat. This guy's got even more double bonds. So we call him a polyunsaturated fat. Either way, it's the same thing. Animal source, generally. Plant sources, generally. A little bit healthier for you, probably. So, fats. Fat is a lipid that has 1, 2, oftentimes 3 fatty acid tails attached to a glycerol. If it has 3, we call them a triglyceride, glycer from the glycerol, and the tri from the 3 fats. So these guys are neutral fats. They have 3 fatty acids attached to that glycerol, and these guys are the most abundant energy source in vertebrates. So the fat that we store on our bodies are triglycerides. The fat that we eat in bacon is triglycerides. A lot of energy there. Maybe not the healthiest thing to eat all the time, but there's a lot of energy there. We concentrate our triglycerides in adipose tissue or fatty tissue. For animals, this is important. It's insulation, right? There's a reason that animals that live in really cold environments have blubber. You know, that layer of fat will keep them warm. Fat also helps cushion joints. It has a lot of good jobs for insulating against the weather and things like that that keep animals alive, helping them survive long periods without food. You can digest your fat until the next meal comes along. So here's our triglyceride. We have a glycerol head, which really isn't that glorious. It's just 3 carbons and an oxygen here, here, and here. And then what we do is we take these really long fatty acid tails, which we learned earlier. We've seen this guy before. We said, why do we have the acid in fatty acid? Because of the carboxylic acid. So we just pulled the hydrogen off this guy and plugged him in here with condensation. We did the same thing with this one and the same thing with this one. And we've got a triglyceride. So when you look at this, it's a really compact group of carbons. That's a lot of energy. For the same reason there was a lot of energy in starch. The same reason there was a lot of energy in glycogen. The only difference is now we can fit so many carbons so close. We can get a lot of energy out of fats, where we couldn't with carbs. So this is a super important lipid. I mean, all the lipids we're talking about are important, but when it comes to like the basic stuff that holds us together, these guys are the winners. They are called phospholipids. The name tells you a lot about what you're talking about. Phospho should remind you of the functional group we talked about a couple lectures ago called the phosphate group, which was PO4. So a phospholipid is a lipid with a phosphate attached to it. Easy enough. So these molecules have a polar head. All of those oxygens in the phosphate group make that molecule polar, separation of charge. And they have nonpolar fatty acid tails. The heads are hydrophilic. Why would something that's polar be called hydrophilic? Hydrophilic means water-loving, and if it's polar, you might recall from chemistry or like a general science the saying like dissolves like. If water's polar and the head of a phospholipid is polar, then it will go to water easily. Now, these nonpolar tails are hydrophobic, water-fearing. And so because they're nonpolar tails and water's polar, they want to get as far away from water as possible. Now, this is really important for us because these guys make up cell membranes and they form what's called a lipid bilayer. And a lipid bilayer means two layers, is going to assemble with all of the polar heads facing the water, the non-polar tails facing away from water. So they give us a two-layer membrane that will contain our cells. And these guys are super important, and I'll show you a little animation of them so we can understand how they work.
Did you know that cells cannot exist without phospholipids? This is because phospholipids form a bilipid layer that make up cell membranes. Cell membranes play an important role in controlling what goes in and out of a cell. Let's examine the structure of a phospholipid. There are four major parts. First, there is a glycerol backbone, as in all lipids. One end of the glycerol molecule is bonded to two nonpolar and hydrophobic, or water-repelling, fatty acid chains. Often, one carbon chain is saturated with hydrogens. In other words, there are no double bonds. But the other carbon chain is unsaturated due to one or more double bonds that give the chain a bent shape. The other end of the glycerol is bonded to a negatively charged and hydrophilic, or water-attracting, phosphate group. Finally, the phosphate group may be bonded to another organic polar molecule, such as choline. Let's now examine.
Okay, so I want to pause it right there so you can get an idea of the whole picture. Hopefully this makes sense as you look through. Surrounded by hydrogens, no separation of charge, so it's non-polar. As we move up to the head, glycerol and the phosphate group, we get all these spare oxygens. Very electron greedy, very electron negative, so they're going to be polar. So we get a polar end, a non-polar end, so we get a water-loving hydrophilic head, and a water-fearing hydrophobic tail. Just want to be sure that this up here matches up with this down here.
How the overall structure of a phospholipid fits its function in cell membranes. If you look at a cell, you will see that it is surrounded by an aqueous environment on the outside and the inside. Focusing on the membrane again, we see it is made of two layers of phospholipids. If you were to drop phospholipids in water, this is how they naturally assemble. The hydrophilic phosphate heads are on the outer surfaces of the membrane bathed in water, and the hydrophobic fatty acid tails make up the interior of the membrane, where they are excluded from water. So you can imagine how early cells might have formed from lipids organizing themselves into a bilayer to create a membrane-bound droplet. In this early cell, the hydrophilic or water-attracting heads form the most outward membrane layer, with the hydrophobic tails facing inward. All you need is a special lipid and water.
Okay, so as far as its function, hydrophobic and hydrophilic ends assembling into membranes, we do know that that works and it helps keep our cells alive, which is super cool. As far as the first mistakenly made lipid from simple building blocks floating around in a pool becoming the first cell, we can certainly discuss the validity of that idea in class if you'd like. Now's not the time, I suppose, to do it because I'm just sitting by myself talking to a computer. But if you want to talk about the evolution of the first cell, we definitely can. As far as right now, I don't know if that's enough to really impress me, but we'll talk about it later. Do understand how phospholipids shapes determine their function and how incredibly vital they are for our cells. That is very important. So there's phospholipids. Hydrophobic tails sandwiched between hydrophilic heads. And here is the picture you guys just saw. It's just a little different structure instead of continuing on to the top like they did before. So our polar phosphate groups, our nonpolar tails, and we have hydrophobics and hydrophilic, and we are set to make a membrane. So a lot of times in class we just draw like a circle with two legs and we call it good enough because it's hard to draw the phosphates and stuff. But that's the idea, is we can assemble these all side by side. And it would look something like this. So one layer of lipids facing the water, let's say outside of the cell. These guys all hiding from water. And then these guys forming the membrane facing the water inside the cell, perhaps. So these guys give us the name phospholipid bilayer. Makes a lot of sense when you look at it this way. And before we just called it cell membrane when we were like, sophomore biology or whatever, and now we understand exactly what makes up that cell membrane. So let's move on to waxes. Here they're giving us the idea of beeswax, forming this honeycomb out of a wax he's making there. But waxes are really useful for a lot of different organisms. They're really complex mixtures with super long fatty acid tails bonded to long chain alcohols. And they're important because waxes are really good at repelling water, and they're really good at protecting structures. Wax is nasty to chew through. Okay, you and I don't eat a lot of foods covered in waxes, and plants will coat their leaves in a thick layer of wax because it's difficult for bugs to chew through it, and it will keep them dry, or rather, I guess it'll keep water from leaving the leaf, if that kind of makes sense. In a dry environment, the plant can survive with a thick, waxy cuticle. It'll keep that water inside that leaf. And so there's reasons to have waxes for building structures, protecting leaves, keeping leaves hydrated in dry environments, things like that. They're very useful. And they're still a lipid. They're still a type of lipid or kind of like a fat. So here's the last one. It's really hard to wrap your mind around being a lipid because we don't associate steroids with fats. We just don't in our heads. But steroids are a type of lipid. Not all steroids are like bad for your health, bodybuilding kind of steroids. We have all sorts of different steroids in our bodies that do a lot of different jobs. And these are lipids. They have a rigid backbone of 4 carbon rings, and they have no fatty acid tails. So as far as energy storage, not really, no. As far as communicating changes in bodies, like endocrine system point of view, absolutely. These guys are super important. Cholesterol is actually a steroid, which is weird because everyone's so worried about the cholesterol levels. Cholesterol is a component of cell membranes, eukaryotic cells. So not bacteria, but everything else, cholesterol's in it. It's remodeled into bile salts, vitamin D's, steroid hormones. We can make estrogen out of it. We can make testosterone out of it. We can build a lot of stuff out of cholesterol, and some of it's good. Vitamin D, that's a good thing. So here's some steroids that we've built out of cholesterol, and you can see that they're really not that terribly different. If you look at estrogen, we've got a couple double bonds here that we don't have in testosterone. Our alcohol group is now a double bonded O. This alcohol group is a double bonded O in estrogen. Estrogen has one methyl group, testosterone has two. By and large, they're pretty similar, actually. I mean, move a couple double bonds around, add a methyl group, and you've completely changed everything about what it does. That's how tightly regulated structure determines function is when you look at molecules and their jobs. So we haven't changed a whole lot, but in the grand scheme of things, we've actually changed everything. So here's estrogen and testosterone in ducks, right? They picked a super colorful duck to exaggerate the point. It's called a wood duck. If you want to see them, go to Canyon Lake. They're not that glorious, but there they are. Female wood duck. She blends in pretty well. She has more estrogen than testosterone. She's about the same size as the male. She might be a little bit smaller, but you can see she blends in well. She could go hide in like some cattails or bushes and be totally fine because she's just brown and white. It's really not that bad. So the male has more testosterone than estrogen, and you can see the changes in his body are dramatic compared to the female. He's got a lot of coloration on his face. He's got a lot of coloration on top of his body. He can't really hide anywhere, but he can attract a female. So there's a job for that specific steroid, but you can see the very dramatic differences that alcohol groups and a couple methyl groups can make on an organism. It's sort of amazing when you think about how small that change is chemically, what a big change it is in an animal. So, take-home messages for lipids. What are lipids? They're fats, they're waxes, or they're oils. We use them fats for energy storage, phospholipids for cell membranes, waxes for plants and structures that we can build if we're insects, and then steroids for regulating our bodies. Triglycerides serve as energy reservoirs. Phospholipids build cell membranes. Waxes repel water. And steroids occur in cell membranes, and we can remodel them to build pretty much whatever we want. And so lipids have a lot of uses, and hopefully that was all pretty clear. If not, let me know in class and we'll talk about it. Thanks.